Hi students, in the last video we have discussed the culture methods, streak plate, straw culture method, then stab culture method and lawn culture method. In this video we will discuss another two culture methods, PourPlate culture method and SpreadPlate culture method. PourPlate culture method. Here PourPlate culture method is mainly employed for enumeration purpose. What is meant by enumeration? Enumeration means counting. So in some situations we need to count the number of organisms or number of bacteria or number of microorganisms in a particular sample. Suppose for checking the quality of a food or quality of water, quality of soil, we have to count the number of organisms, or we need to understand the actual number of live bacteria, the bacteria or the microorganisms that are live or living, not dead. For that, the most important method used or commonly used method is PourPlate. So the aim of PourPlate is to enumerate microorganisms or bacteria in a sample. The sample may be a soil sample or water sample or food sample or any sample. So if you want or if you need to understand the number of organisms, the number of living organisms, here we are discussing the bacterial culture. So the number of living bacteria, the important method or the routinely used method you can adopt is PourPlate method. For isolation also this method can be used. Okay, for isolating bacteria this method can be used, but mostly this method is used for enumeration purpose, enumeration of microorganisms present in a sample. Okay, then we will check how we can do this. Okay, in your higher semesters you will study, you will have the practical sessions of PourPlate. Okay, at that time you will understand more, or you will do this by yourself. Okay, here different types of or different numbers of tubes are required. So tubes containing 50 ml of the agar medium are melted and left to cool in a water bath at 45 to 50 degrees Celsius. Okay, here we need in two ways we can do this, but the most commonly used method I am explaining, the most routinely used method. Here first you have to prepare the agar medium in tubes. Okay, in test tubes you have to prepare the 15 ml of the agar medium or 15 to 20 ml of the agar medium. You have to prepare in tubes and you have to sterilize it. Then after that you have to cool it. Okay, you have to cool it by keeping it at a water bath set at 45 to 50 degrees Celsius, because we have to pour. After that we have to pour. Okay, otherwise if the temperature is fall below 45 degrees Celsius, it will become solid. So we cannot pour the content from the tube into to the petri dish. So first we have to prepare that. Then different dilutions of the sample were made by serial dilution technique. The next step for PourPlate culture is you have to prepare different dilutions of the sample. The sample may be, so for example, suppose we have to enumerate the number of organisms in a soil sample. So first we have to prepare the soil. You have to take the soil sample and then mix in a suitable amount of sterile water, then thoroughly mix it, and then you have to dilute it. You have to make different dilutions of the sample. That technique is called the serial dilution technique. Serial dilution technique. That is, this step is very important for PourPlate technique. So we need 15 ml of the agar medium in test tubes kept at 45 to 50 degrees Celsius. You have to maintain a liquid what condition, okay, by keeping it at 45 to 50 degrees Celsius water bath. Then you have to make different dilutions of the sample by what method? Serial dilution technique. Next we will discuss what is serial dilution technique. It is very simple. We have to make different dilutions. We have to dilute the sample. Suppose but the number of organisms is very huge. At that time we won't get, after the plating, we won't get the well separated colonies, so that it will become difficult for counting. We can't count. So we need well separated colonies on the agar surface or agar medium. Then only we can count the number of colonies. So for that we have to dilute the sample. Anyway, you have to dilute the sample. Okay. So I will explain how the serial dilution technique is performed. Here very simple. We need 9 ml of sterile saline. Okay, the saline is commonly used. Sterile saline is used, or sterile water. We can use sterile water also. 9 ml of the sterile. You have to make different in different tubes 9 ml sterile saline or sterile water, or you have to pour it the saline or water. Then you have to sterilize it by using autoclave. Okay. Okay, then for first dilution, that is the first dilution is 1:10. How we can make 1:10 dilution? Just take from the sample, you have to take 1 ml of the sample by using a sterile pipette. Okay, by using, everything should be sterile. So by using a sterile pipette or sterilized pipette, then you have to transfer 1 ml of the sample from, we have already prepared 9 ml of the sterile saline tubes. So take one sterile 9 ml saline tube and then add this 1 ml to the 9 ml of the sterile saline tube. So it makes 1:10 dilution. That is 1 part of the sample in 9 ml of the water. So 1 in 9 or 1 by 9 or 1 in 9, not 1 in 9, the total 10, the volume is 10. Then out of the 10 ml, 1 ml is the sample and 9 ml is the saline. So it makes 1:10. In 10 ml, the 1 part is the sample. So it makes 1:10 or 1 by 10 or 1:10 or 10 raised to minus 1. 1 by 10 is equal to 10 raised to minus 1. So the first dilution you made, you just take 1 ml sample and then add to 9 ml sterile saline. So it makes 1:10 dilution or 1 by 10 dilution or 10 raised to minus 1 dilution. Okay. Then the next dilution, that is 10 raised to 1 in 100 dilution. How we can make 1 in 100 dilution? Next is tenfold dilution. It's tenfold dilution, not double dilution. Here we are making tenfold dilution. For making 1 in 100, next after 1:10, next is 1 in 100. How we can make 1 in 100? So it is already 1 in the first tube is, here it is 1:10 dilution or 10 raised to minus 1 dilution. Take 1 ml from the 10 raised to minus 1 dilution and add this 1 ml to the next 9 ml of the sample. So it is already 1 by 10 diluted sample. We are adding it to 9 ml. So total 100 in the 1 in 100. The total makes 100. 1 in 100 or 1:100 or 1 by 100 or 10 raised to minus 2 dilution. So how we can prepare 1 in 1000 dilution? Take 1 ml from 10 raised to minus 2 dilution or 1 by 100 dilution and then add it to next 9 ml saline. So it becomes 1 in 1000. Like that we can prepare 1 in 10,000 or 1 in 1 lakh. That is 10 raised to minus 1, minus 2, minus 3, minus 4, minus 5, minus 6, minus 7. Like that we can prepare the dilution of the sample. And this technique is called the serial dilution technique. Okay, so now we have 15 ml agar medium in tubes and also different dilution. That is, we have prepared the sample dilution from 10 raised to minus 1 minus 2, minus 3, minus 4, minus 5, minus 6, like that. Okay, minus 7, we can prepare up to 7 or 8 depending upon the concentration of organism in our sample. Okay. Okay, then from each tube we have prepared 1 in 10, 1 in 100, like 10 raised to minus 1, minus 2, minus 2. From each tube take 1 ml from this dilution and transfer it to the molten agar in tubes. We have different agar, different tubes having 15 ml agar. We have already kept in water bath. Take 1 from that and then you have to add 1 ml from 10 raised to minus 1 dilution. Then shake it, mix it properly and then immediately pour into a sterile petri dish. Allow it to set. Okay, then keep it to set. Then take next tube, next dilution, 10 raised to minus 2 dilution. Take 1 ml from by using a sterile pipette and then transfer it to another 15 ml of agar tube and then mix it, pour it into another sterile petri dish. Like that you have to complete all the dilutions. Okay, take 1 ml from all the dilutions and then you have to add the respective tubes and then you are, after mixing in agar, then you have to pour the corresponding petri dishes. Now we have different dilution or different agar plates having different dilutions of organism. Then after complete setting of the agar, you have to keep it in the incubator for incubation for 24 to 48 hours. Okay, normally for 24 hours, if it is a soil sample, we have to wait for 48 hours, 2 days, 1 to 2 days, 24 to 48 hours. Then after incubation, the colonies will be seen well distributed throughout the depth of the medium and can be enumerated by using our naked eye or by using a colony counter. In our lab, we have a digital colony counter. By using that, we can count, or by simply we can count. Okay, by observing the plate, we can count. By using a pen, we can or marker pen, we can count. Okay, that's the method adopted. That is when the dilution increases, in that plate we can expect the well separated colonies. Okay, that means in 10 raised to minus 4 or 5 or 6 we can see well separated colonies. Then that plate can be used. Normally the plate which is taken for the counting is, we have 10 raised to minus 1, 2, 3, like that. We have different plates after incubation. In front of us we can see different plates. So how we can select the plate which is apt for counting? There is a criteria, that is a plate containing 30 to 300 number of, minimum it should contain 30 number of colonies and maximum 300. Okay, if it is more than 300, we can we can't count that, we can't use that plate for counting. Or if it is less than 30 colonies, it should not be counted. So the plate should be selected which contains 30 to 300 number of colonies. The minimum should be 30, the maximum 300. Okay. Then after, suppose we counted this 280, suppose it is 150, we got 150 number of colonies in 10 raised to minus 5 dilution. So we can convert it into a colony forming unit. That is colony forming units means CFU. CFU, colony forming unit, that is the number of organism is expressed in terms of colony in the unit, colony forming unit, CFU per ml, colony forming unit per ml of the sample. How we can calculate CFU per ml of the sample? It is number of colonies counted into the dilution factor. If it is 150 in 10 raised to minus 5 dilution, we got 150 colonies. So the CFU per ml of the sample is 150 into 10 raised to 5 CFU per ml. The unit is CFU per ml, colony forming unit per ml of the sample. Okay, it is not colony or it is not an organism, it is colony forming units. Because why we are using CFU, colony forming units? Because the bacteria, a single colony is developed from a single cell, not only from a single cell. Suppose in some situations the bacterium will be in chains or in pairs. So a colony is produced from, may be produced from a single cell or from a group of cells. Okay, a chain of cells. So that we are expressing it as CFU, colony forming units. The unit may be a single cell or a group of cells. Okay, or two cells or three cells or four cells. Okay, depending upon the type of organism. That's why it is expressed as CFU, colony, not colony per ml, colony forming units per ml of the sample. Okay, I think you have understood. Okay, and this is a technique. You can see this is a technique. It's a diagrammatic representation of the technique. That's the original inoculum. That is the first sample. You have to add, if it is a soil sample, you have to add 1 ml, or it's a water sample, you have to add 1 ml. Then from the 1 ml, you have to add, is the 9 ml tubes. Okay, different 9 ml tubes. Okay, of the saline, containing saline, sterile saline. You have to take 1 ml and then transfer to 9 ml saline. Becomes 1 in 10. So it is 1 in 100. Then 1 in 1000, 1 in 10,000, 1 in 1 lakh, like that. So after plating, you have to take 1 ml from each tube and then pour into this. Okay, you will get like this. Not you can't you won't get what isolated colonies. So this can't be used for counting. So here also we cannot we won't get single colonies. Here also it is not single colonies. It is more than 300. Then in 1:10,000 dilution, we got that we can count 1, 2, 3, like that we can count. But then you have to note down how many colonies into its dilution factor. Here also it is 1, 2, 3, 4. So we cannot use this plate. So this is the apt plate for counting. So you have to count 1, 2, 3, 4, 5, 6, 7, 8, like if it is 80, then 80 into what? 10 raised to, it is 1 in 10,000. Okay, 10 raised to 4. Like that, not 10 raised to minus 4. So dilution factor 10 raised to 4. Okay, not 10 raised to 1 in 150 into 10 raised to minus 4. Okay, it is not, it is we won't get an exact number. It is 0.000, like that. So it is 150 into number of colonies into dilution factor. The factor is always positive, 10 raised to 4 or 10 raised to 5, like that. Okay, the calculation number of colonies on plate into reciprocal of the dilution of the sample. For your understanding, reciprocal of the dilution of the sample is equal to number of bacteria per ml. For example, if it is 32 colonies are on the plate, it is 1 by 10,000 dilution, then the count is 32 into 10,000. That is 320,000 bacteria per ml in that sample. Okay, clear? So this is another diagram showing the PourPlate technique only. Okay, the next is SpreadPlate. Here SpreadPlate is also similar, but here also we need a serial dilution technique. That's why I am explaining the PourPlate and SpreadPlate at the same time. Okay, so SpreadPlate only we can see some differences. Okay, for preparing SpreadPlate, appropriate dilution of the sample is prepared by serial dilution technique. Here also it comes serial dilution technique. If you have to serial dilute the sample. Okay, like 10 raised to minus 1, 10 raised to minus 2, 10 raised to minus 3, like that you have to count, you have to dilute it, dilute the sample first. Okay, here only thing is after that, but only point we need an agar in tubes or agar in not in tubes. Agar here in the previous technique, that is a PourPlate technique, we are using agar molten the agar in tubes. But here first you have to prepare the agar in petri dishes. That is approximately 20 ml of the agar is poured into petri dishes, and then you have to make the a uniform agar surface for the spreading of the sample. Okay, like we need an agar plate which is we previously used, that is in the streak plate we need an agar plate. Like that here also we need an agar plate, a well dried agar plate is required, you have to prepare that. We have to prepare that first. Okay, and after that you have to make the seal dilution. Then take 0.1 ml, not 1 ml. In the PourPlate we are using 1 ml. Here we are taking from the sample, diluted sample, we are taking only 0.1 ml and distributed the sample uniformly over the agar surface by using an alcohol dipped flame sterilized glass spreader or plastic spreader. Okay, or any sterile spreader can be used. Okay, take 0.1 ml from the diluted sample and then place it over the agar surface. Okay, by using a pipette, transfer 0.1 ml from the sample and then place it over the agar surface. And this sample is uniformly spread by using a alcohol dipped flame sterilized. This is a method of sterilizing the glass spreader. Okay, first you have to dip in alcohol, then you have to show on the flame. So after that, after cooling the glass spreader, you have to spread the sample all over the agar surface. Then you have to place it for incubation in incubator for 24 hours. Okay, 24 hours we have to wait. Okay, then after 24 hours we can see, depending upon the dilution, we can see the development of well isolated colonies. So this colonies, from this colonies we can pick the one which is required for us. Okay, or required for you. Okay, so the well separated colonies can be seen on the agar surface. So this method is employed for isolation, isolation method. That is like streak plate. This method is also used for the isolation of the pure culture. Okay, this method is employed for isolation of bacteria from soil sample, water sample or food sample. Okay, the streak plate is used for isolation of bacteria, especially from clinical samples. Here this spread plate is mainly employed for isolation of bacteria from soil or environmental samples like soil sample or water samples, then also food samples also. This method is the important, this what most suitable one. So this is a method, the diagram shows this is a diluted sample. Okay, diluted sample, you have to transfer we are transferring 0.1 ml, pouring 0.1 ml and then you have to spread it over throughout the, okay, throughout the this one. Okay, so you will get the well isolated colonies over. It's on the surface you will get only the surface growth, you will see the growth only on the surface. In the spread plate method, this is it's a comparison of PourPlate and spread plate. Here this is a PourPlate. If it is a PourPlate, you have to mix the sample first. Okay, here first we are transferring from the dilution, we are transferring directly on the agar surface in spread plate. But in PourPlate, first we have to add the sample to the agar medium which is in tube. Then we have to pour. So in spread plate we will get only surface bacterial growth. So we can easily isolate. So in PourPlate we will get the growth on the surface and also inside the medium, because we are mixing, before pouring we are mixing the sample with the agar. So we can expect the growth inside the agar also. But in spread plate we will get the growth only on the surface. That's the difference. So that it's used for isolation and this is used for, PourPlate is used for enumeration. Thank you.